Today I am going to discuss a topic that is nucleic acid. Now basically there was a scientist whose name was Friedrich Mischer. First of all, there was scientist Friedrich Mischer he basically observed a chemical composition and a nature of the pus cells. Basically, there was a scientist whose name was Friedrich Mischer who observed the chemical composition and the nature of the pus cells. He observed that the pus cells are basically the dead white blood cells that are present on the infectious part of a body. So he observed that the pus cells are quite different. They are quite different from the normal cells. So what he did, he gave the name of pus cells as a nuclein. Then further he studied about the composition and the nature. Then again he observed some acidic nature, acidic properties. Then he renamed that nuclein as a nucleic acid. That's how that first of all he observed he was studying about the chemical composition and the nature of pus cells, and then he observed that the pus cells are quite different from the normal cells, so he named them as a nuclein. Then again he further studied and he observed some acidic properties into it. Then he renamed that nuclein as a nucleic acid. Then further investigation was done and it was observed that the nucleic acid is basically consists of three things. First thing is pentose sugar. Second, he observed nitrogenous bases. Third thing, he observed phosphoric acid. Three things he have observed. Then further study was done on pentose sugar. He observed two sugars into it, ribose sugar and second was deoxyribose sugar. Three things. Ribose sugar basically contain CH10 and O5, and in deoxyribose it contain C5H10 and O4. That was observed in pentose sugar. Then he studied further about the nitrogenous bases. Nitrogenous bases basically consist of two bases. It is divided into two bases. First is purine base, and second is pyrimidine base. Basically purine, there are five bases, different bases, that is adenine, guanine, cytosine, thymine, uracil, that was observed in these two bases. In purine bases consist of two bases, that is adenine and guanine. And pyrimidine base consists of three bases, that are cytosine, thymine and uracil. So basically these were three bases that was in the pyrimidine bases. So these are five bases, but it is divided into two bases, purine bases and pyrimidine bases. And phosphoric acid, third thing he observed, that was phosphoric acid into a nucleic acid. Now how can we identify that which one would be guanine, adenine, cytosine, thymine or a uracil? So let's talk about their structures. If we talk about a purine base is basically consists of the two rings. the difference between them, we can differentiate that purine base is consist of two rings, means either adenine or guanine. They both will be having two ring structure, and pyrimidine bases will be having one ring structure. For example, these are, and here two rings are there, so one ring will be hexagonal and second ring will be pentagonal. Okay, so if we talk about adenine, the to identify adenine, there is a trick that NH2 NH2 means amine group will be attached on the sixth carbon. If amine group is attached on the sixth carbon of the double ring structure, then this ring structure would be known as adenine. And if there will be a double ring structure and amine group is attached on the second carbon, amine group is attached on the second carbon, so that double ring structure will be known as guanine. That can easily, we can identify that which one is adenine or guanine. So there adenine or guanine, there will be a double ring structure because they are purine bases, and if the NH2 means amine group is attached on the sixth carbon of the double ring structure, so it will be called as adenine. And if it has a double ring structure and amine group is attached on the second carbon, so it will be known as guanine. Now these three, basically by seeing them, we can identify that these are single ring structures. So these are basically pyrimidine bases. These all are pyrimidine bases because they have a single ring structure, and purine bases have double ring structure. So if I talk about thymine, how can we identify thymine? If CH group is attached, wherever it is attached, but on the top, we can say a methyl group is attached with the single ring structure, that will be known as thymine. Then if NH2 means amine group is attached on the top of a single ring structure, that will be known as a cytosine. But similarly, if there is also a pentagonal structure, means a double ring structure, that will be known as adenine. But it is a single ring structure, NH2 is attached on the top of the single ring structure, so that will be known as cytosine. And uracil, neither having a CH3 group or neither having a NH2 group, means both groups are absent in this single, it will means it is a simple single ring structures, that will be known as uracil. And basically uracil is present in RNA, it is absent in DNA, and thymine is present in DNA, not in RNA. So these three are basically the pyrimidine bases and having a single ring structure. And if we talk about purine bases, they are having a double ring structure. And this first ring will be hexagonal and second ring will be pentagonal.